My name is Kevin S. I'm the Chief of Pediatric Neurology at Vanderbilt Children's Hospital. I also direct our alternating hemiplegia of childhood clinic there, and we've been doing that for the last 4 or 5 years. I also have a fairly extensive basic science research effort into AHC-related diseases. Here at the 7th International Symposium for ATP1A3 disorders in Chicago, Illinois, and it's been a fantastic symposium so far, though I have to be careful because I'm one of the organizers, but I think so far it's been fantastic. And you can really see from previous years how the field has matured. Stories that got started a year or two ago are developing. We have more answers. Of course, that leads to more questions. But it's also very exciting as we're seeing more and more new people come in to this field. And we're also seeing, as before, parents are very important and asking the really perfect questions. I think this is a really well-working process, and I really, again, look forward to the next year meeting and beyond that. So this is a really fast-moving but exciting field. But I recognize that for families in particular, it never goes fast enough, and things we do, while exciting they agree and making advances, will help their child today, tomorrow, and next year. And it's hard to know, but I think we're starting to see, again, movement in those directions. But I hope we can accelerate the process of all of that. So my research interest, and which was discussed by one of my students, John Snow, at this meeting, was really building on the stuff we presented the last couple of years, which is to make stem cells from patients with AHC, really well characterize these cells, meaning making sure that they are stem cells, they have all the properties of stem cells, they can make neurons well and other cell types, making sure they have normal chromosomes, which can change when you actually make these stem cells from patients' skin cells, which is how we obtain them. And then also to verify that they still have a mutation, usually in the ATP1A3 gene. That's kind of a basic thing. But sometimes you can make stem cells, and along the way, you're no longer studying the disease model. So all that's quality control, essentially, but it's really difficult quality control. And the protocols are very laborious. So we made good progress making many different lines from patients with most of the major mutations, and now we're going through and making stem cell lines from patients with the less common mutations. But again, I think those are going to be very important to help us understand what the protein function is in this disease. And then the next steps is also the hardest, which is then once you have stem cells made from patients and they're well characterized and they do all these things, making cells to study is very hard. So making human neurons from human stem cells is definitely possible. We've done it for years with our close collaborator, Dr. Al George. We published a paper in the last year showing some of the electrical changes in those cells, but getting enough cells and the right type of human neurons is just an enormous challenge. But I remind people this field of making stem cells from patient cells did not exist 10 years ago. And we've gone from no approach at all to studying human neurons to having a really well-developed system in a dish where we can make these cells and study them. So again, the pace of discovery is never going to be fast enough for some people, but it's already tremendously fast, at least compared to some previous ways we've done things. So the research efforts now are really trying to understand better what is the impact of the various mutations in the alpha-3 function. We're trying to look at that through expression patterns, as other genes are expressed. When a neuron has a change in its alpha-3 protein, what does that do to that cell? Does it change other genes to compensate for that deficit? We're starting to do some electrophysiological studies to see what, again, is the impact of the mutation on the electrical firing of the cells. And these are really complements, again, to what our other collaborator, Dr. Al George's lab, does in terms of doing electrophysiology. So we're moving faster, we're coning down the activities and focusing on different cell types and definitely making progress. And we hope as we move forward, this will lead to, again, new insights into how these genes and cells misbehave, and then obviously how that would then lead to finding better and better therapies to stop the symptoms as quickly as possible and even reverse some of the abnormalities that patients see. Sure. I think I alluded to already that I see tremendous progress, and the pace is actually accelerating and compared to other disorders I've worked with, and where we're at in 2018 for anything versus where we're at in 2006 and before is incredibly fast. But no, I'm not satisfied with the pace because it's not fast enough to help the patients we have right now. And if another child is diagnosed tomorrow with AHC-related condition, I don't have a lot of tools to offer them. And I think there is really much a race against time. And in doing research, we understand this, and the families understand it, but I know they need answers now. So I hope it really does accelerate, and I hope we have much better therapies. So yeah, it's a mixed question, but I'm never patient enough to say yes, it's all good, because we're not delivering soon enough. It's always, yeah. So if other way of asking a question is, if you had unlimited funds, would that, make things go faster? Yes, it would. But I think there's still some fundamental things we don't understand yet about what AHC and related disorders really are, and that will just require maturation of technologies, maturation of our thinking processes and things like that. But more money means more people working on it, and you can catalyze probably and make it process. So I think that is the bottom line. What can we ask for now? And I'm not asking anyone in this organization here or who's sponsoring ready to do it for more funding, because they've already been very generous. But having more governmental funding and having NIH in the United States, for example, grants to really invest more in these efforts would be well worth it. And one of the things that's been terrific about the meeting this year, as well as in years past, we've had very good representation from very senior people at NIH come to this meeting, hear what we're trying to say, and then we can use that to have a back and forth. And they definitely want to support the best possible science. And then our job is to present them with the best possible science to then go forward. So yeah, funding is a key part right now. But again, I think it's just again, maturation of technologies and our understanding of what's going on. And compared to 10 years ago, we're completely different. And I'd like to think the next 10 years will be stuff I can't even imagine what's going to be. But is that fast enough? If you have a 2-year-old child right now, probably not, but we'll see. Yes, I tend to be a very optimistic person in general for these kind of things, because again, during the course of my career, just seen tremendous changes, and that should continue, and things that we could never even think about treating. At least I'm talking about now with diseases that affect children's neurologic function. We can now start to treat them in very fundamentally different ways. So yes, I'm optimistic, and I think that's the way to be as you approach these very difficult problems. But again, there's real technological advances and examples in other diseases that we can learn from that underlie that optimism. I mean, it's a wonderful international community. Them being present and just speaking to challenges they face is inspiring and helps us stay grounded to what we're trying to do. I guess if you had concrete examples of what they could do, gets back to my earlier comments, more lobbying to their local governments to support this research is much more powerful when they hear it from an affected family, I think, than it is to scientists, since obviously we're the beneficiaries of the research funding. It's our careers. But they understand that we're pushing for these things for the right reasons. But I think coming directly from families, and I think the partnerships we've had here have been terrific. So I think just continue doing that advocacy part of it and continuing to engage physicians and scientists who work in AHC-related disorders is key. And I will also say, I don't think this group has this issue, but I wouldn't be shy about challenging us. Why do you think this? And how will this help my child now? And we really enjoy that dialogue, because that's not something we get often from other families. I think this is just, again, a very well-educated community who is pushing hard for all the right reasons. So I think continuing what we're doing is basically what I would recommend, and maybe, again, kick it up just a little bit if you haven't already in terms of lobbying to your local governments for increased research funding so we can make that next breakthrough faster.